Well, let me begin by thanking you for your invitation to address your Congress, and in particular to focus my remarks on one of my scientific heroes, Dr. George Palade, whom you see seated here next to his favorite instrument, the electron microscope, which he used to great advantage to examine the interior workings of cells, particularly cells of the pancreas. He was an inspiration not only to me, to many young scholars in cell biology, starting in the 1950s and 60s, and when I became involved in research on the process of protein secretion in the mid-1970s. I was inspired not only indirectly, but very directly by Dr. Palade, as I'll indicate in my talks. So let me begin by telling you briefly about his achievements and what we learned by his use not only of the electron microscope, but also of the techniques of self-fractionation that allowed us to separate and to examine in molecular detail some of the organelles that interrelate by the process of protein secretion. As I'm sure you're all aware, Dr. Palade received the Nobel Prize for his work on dissecting the process of protein secretion in the exocrine pancreas. And he put together the highlights of his work in an article published the following year in Science magazine, where he described the state of cell morphology when he began, and then as he explored using microscopy and self-fractionation to reveal the interrelations of organelles in the secretory pathway. Dr. Palade and his colleagues, particularly Dr. David Sabatini, were able to perfect techniques that allowed the preservation of samples, fixed tissues, allowed their preservation so as to resist the forces of the electron beam that is projected through a vacuum chamber to reflect on particles and membranes in thin sections of plastic embedded cells. And here are some of those really beautiful and really unmatched morphologic inspections of cells of the exocrine pancreas. On the left, you see a thin slice through the cytoplasm, revealing this tubular network of membranes called the endoplasmic reticulum. Palade did not discover the endoplasmic reticulum. That was actually discovered by Keith Porter, a colleague of his at Rockefeller University. But the precision of his analysis allowed us to visualize ribosomes, these dark staining structures on the cytoplasmic surface of the ER membrane. These are ribosomes that Palade himself discovered in the mid-1950s and was able to show that these are the particles that organize the production of polypeptide chains. In Palade's work, he showed further that these ribosomes engage in the synthesis of proteins that are going to be secreted from the pancreas, proteins such as insulin, that are made by the ribosomes and vectorially transported across the membrane of the endoplasmic reticulum into the luminal space, the interior space of the organelle. And you can see from this slice on the left that this network is an elaborate network, almost like a canal network, where the luminal space of the ER is all continuous, and along which proteins that have passed across this membrane fold and then are packaged for secretion to the next stage in the pathway. He showed, using isolated membranes with ribosomes still bound, that it was possible to complete the synthesis of secretory proteins that would then fold and form disulfide bridges in the luminal interior of this organelle. And this as a prelude to the selection and capture of molecules for packaging into vesicles that are transmitted step by step through membranes called the Golgi apparatus. These Golgi membranes had been described by an Italian neuroscientist, cytologist by the name of Camillo Golgi in the 19th century. They were found ubiquitously in all tissues, but it was up to Dr. Palade to demonstrate by a beautiful series of experiments that molecules progress from the ER into this structure, the Golgi apparatus, And finally, with time, these molecules progress into immature and then into mature secretory granules. He did this by the use of autoradiographic incorporation of radioactive amino acids into proteins, followed by a period of incubation so that the molecules would move from one station to the next, and finally revealed by capturing thin sections on a photographic emulsion where the C14 decay of radioactive amino acids would register as the location of a protein during a pulse and a subsequent chase. This elaborate and technically very demanding series of experiments allowed him to demonstrate that the proteins passed from one stage to the next over the course of about 30 to 45 minutes on their way to be secreted from the exocrine pancreas cells. Another example of a cell that he studied in some detail, but which is of a picture shown by a latter-day protege of Dr. Palade's, Dr. Lelia Orci. This is a thin section of the beta cell of the pancreas, showing the elaborate accumulation of granules containing a nearly crystalline form of insulin sequestered within the interior of a lipid bilayer, the membrane of the granule. And from Palade's work, we know that these granules accumulate proteins in advance of secretion, and that in a timed and controlled manner, these granules move to the inner surface of the plasma membrane of the cell, where a process of membrane fusion allows the granule to be discharged to the cell exterior. And in this case, the crystalline insulin dissolves and is distributed into the bloodstream. So all of these events were captured by the microscopy of Dr. Palade. Now, this process extends far beyond the pancreas and includes virtually all the cells, all eukaryotic cells, and all of the cells in the mammalian body. One area where this process is especially important is in the brain, where other kinds of granules, in this case, small membrane particles called vesicles or synaptic vesicles, organize for the secretion of chemical neurotransmitters that are discharged by a process of fusion of this membrane at the presynaptic membrane, where the neurotransmitters are then exposed to a space, in this case between a nerve cell and a muscle cell, or in the brain between nerve cells, where the chemical neurotransmitters diffuse through this gap called the synapse and bind to and activate receptors on the opposing membrane to activate new channels that in this case lead to muscle contraction, or in the case of the connection between nerve cells, lead to the transmission of a nervous impulse called an action potential from one cell to the next. So the means by which cells communicate in the brain depends intimately on a knowledge of how these vesicles are produced, how they are delivered to this presynaptic terminal, and how the membranes may fuse at this location. Finally, in summary then, from Palade's work, we knew in the mid-1970s that vesicle traffic mediates the flow of proteins such as insulin or hormones or other molecules like antibodies. These membranes intercommunicate by a flow of vesicles, often vesicles that contain a kind of a coated surface that appears to be involved in pinching a vesicle to collect cargo molecules that are destined for traffic to the Golgi apparatus or to a mature secretory granule. Finally, in his summary statement in this famous Nobel lecture that he delivered in Stockholm in 1974, Palade was offered the opportunity to speculate about the role of his discoveries in other processes, and he quite brilliantly concluded that the process of secretion might also be linked to the assembly of the plasma membrane, whereby a granule with a membrane surrounding it would by fusion not only deliver cargo molecules like insulin to the outside of the cell, but that the membrane itself would become a building block for the growth of the plasma membrane. And so he drew this postulate among the non-secretory functions postulated for the endoplasmic reticulum in the Golgi complex is the production of cellular membranes, plasma lemma or plasma membrane included. At present, this postulate rests only on suggestive evidence, most of it morphological. But he was, of course, quite correct. And what I'll tell you about now in the next few slides is how my work, beginning in 1976, led to a confirmation of that hypothesis and more. Now, I had the pleasure of meeting Dr. Palade when I was a graduate student in the laboratory of Arthur Kornberg at Stanford University. Kornberg was a biochemist far removed from the kind of morphological work that Palade perfected, but Kornberg appreciated Palade's work so much so that the year before I began graduate school in 1969, Kornberg traveled to New York, where he spent several months studying with the members of the Palade laboratory. Several years later, when I began my career at UC Berkeley, I had the pleasure of hosting Dr. Palade for a series of honorific lectures that he delivered, shown here with a picture of a vesicle, a cartoon with a vesicle fusing and delivering cargo. They were the annual Smith, Klein, and French lectures in 1978, just shortly after I began. So it was my great pleasure to speak directly with Dr. Palade and to share with him the excitement of our work using yeast, a simple microorganism, to study the secretory process. And I recall vividly that Dr. Palade was quite surprised that yeast cells manufacture glycoproteins in a process of secretion, at least superficially similar to animal cells. Well, let me just, reflect on something that happened right around the time I met Dr. Palade. He came to Romania for an honorific series of lectures at the opening celebration of the Institute of Cellular Biology and Pathology in the year following my meeting with him in Berkeley. And there are many people who some of you may recognize in the front row, including the Simonescus, who were the founding directors of this institute, and the proteges of Dr. Palade, shown in the middle here, Gunther Blobel, who went on to his own great success and a Nobel Prize, and his one of his co-Nobel laureates standing to his left, Christian de Duve, and one of his younger proteges, also a remarkable cell biologist by the name of David Sabatini. So this was a wonderful occasion. Regrettably, I was not a participant in that particular meeting. Well, let me now tell you about where my work began. As I left my postdoctoral career, I started as a junior faculty member at the University of California at Berkeley with the intention of studying a simple eukaryotic microorganism, Baker's yeast. And I use this image of a cluster of yeast cells, such as you may see growing on the surface of a grape, and contrast it with the painting of Charles Darwin, the father of our understanding of evolution. There is, quite surprisingly, unexpectedly, a remarkable connection between a lowly yeast cell and a human being, because the inner workings of the yeast cell, the activity of the cytoplasm and of the nucleus in the yeast cell, is evolutionarily conserved through 2 billion subsequent years to the present day human race. And there are many experiments that preceded my own work that demonstrated that the genes that yeast cells use to organize themselves are very similar, highly related to similar genes in the human genome. So there is a quite remarkable evolutionary link of conservation of gene sequence and function in spite of 2 billion years of passage. If you have a closer look at a yeast cell, of course, it looks very different than a human cell. Yeast cells grow by a budding process, not by dividing in half. When they are growing in a rich nutrient broth and the conditions are favorable, a yeast cell will create a small new cell called a bud growing on the surface of the mother cell. And during the course of the 90 minutes or so that it takes a yeast cell to divide, this cell gets bigger by the apparent growth of new surface material at the bud surface itself. Finally, when the daughter and mother cells are of equivalent size and where the nuclei have segregated into both cells, there is a division event that is somewhat related to that occurs in mammalian cells to create two completely new cells that can continue to grow indefinitely as the conditions are correct. Now, if you cut through a yeast cell to inspect very closely the interior of the cell, to look at the cytoplasm, you see something very different than an image I showed you a few minutes ago of a pancreatic beta cell. This is a thin slice through a yeast cell, using basically the tools that Palade developed to investigate the mammalian pancreas. Most of the cytoplasm of a yeast cell is filled densely packed with these particles that are ribosomes. They're busy synthesizing polypeptide chains. Of course, the polypeptide chains that a yeast cell uses. There is evidence of organelles similar to those found in mammalian cells. There is, of course, a cell surface, a plasma membrane that surrounds the cell. There are tubules of the endoplasmic reticulum, often lining the inner surface of the plasma membrane of the cell. This is much less obvious or less apparent than the tubular network in a mammalian cell, but nonetheless, it's there. And in some other images, you can see it quite abundant. In other sections, not this one, you can see a cross section through the nucleus. In this case, we see a cross section through a structure in a yeast cell called a vacuole, which is similar to the organelle that we have in our cells called a lysosome, which is an organelle that degrades molecules that are no longer needed. Our attention initially was drawn to a small number of membrane vesicles that appear under the bud portion of a dividing yeast cell. These look superficially similar to the vesicles that I showed you at a nerve terminal. But of course, these are vesicles that a yeast cell makes, and that does not package insulin or package chemical neurotransmitters. These vesicles package yeast cells, proteins that yeast cells make, that it secretes into its environment to the outside of the cell that are used to harvest nutrients that the yeast cell wants for its nourishment. Now, in addition to a role in secretion, we imagined, as Palade speculated in his Nobel lecture, that the membrane that surrounds this vesicle would join the plasma membrane in a process of membrane fusion that would allow step by step the plasma membrane to get bigger during the 90 minutes or so that it requires a yeast cell to grow and divide. So that offered a simple and powerful prediction to us, one that is experimentally tractable using yeast. And that is that the production, the movement, and the fusion of these vesicles at the cell surface would be essential for the yeast cell to grow and divide. If that were so, then mutations in genes required for this process would cripple the cell, would prevent it from growing and dividing, and would thus produce a lethal death phenotype. So that's a powerful prediction. Yeast cells have about 6,000 genes, and about 1,500 of those genes are essential genes. That is, if you make mutations in those genes, the yeast cells can't grow and divide. Now we imagine among those 1,500, there would be some that were uniquely required for the process of secretion, as opposed to other cellular processes such as DNA replication or messenger RNA production. So one of the tools that's available for microorganisms is a simple idea that allows one to isolate lethal mutations and yet to allow the cell to grow under certain conditions. The logic is as follows. If you expose a cell like yeast to a chemical that causes mutations randomly, occasionally a mutation will change just one position, one amino acid in a protein that renders that mutant protein thermally unstable. In other words, the protein may function if the cell is growing at room temperature, but if that mutation causes the protein to misfold or unfold when the cells are warm to, let's say, human body temperature, that would, on elevating the temperature from room temperature to body temperature, produce a lethal phenotype. And this is very common, commonly used. It's possible to isolate such temperature-sensitive lethal mutations, to grow them to propagate cells and colonies that form on a Petri plate at room temperature, and then to culture the cells and to change the temperature to human body temperature, 37 degrees, at which point such lethal mutations start to die. Now, my first graduate student, Peter Novick, shown here in 1978, busy working at his bench, he and I decided to simply look for among temperature-sensitive growth colonies, for those that cause enzymes that would normally be secreted outside of the cell to be accumulated inside of the cell using simple biochemical assays to measure two different enzymes. We found initially a mutant that we called SEC1, short for secretion number 1, which when the cells were incubated at room temperature seemed to grow and secrete normally. But as soon as the cells were warmed up to 37 degrees, these enzymes started to build up inside the cell. Now, fortuitously, at that point in 1978, Dr. Palade, in May of 1978, came for the visit where he gave those honorific lectures that I mentioned. And Peter Novick and the other graduate students hosted Dr. Palade for dinner. And Peter told him about these new results that he had just obtained with a temperature-sensitive secretion mutant. And Palade said, well, of course, now you must examine these cells by thin section electron microscopy. And one of the great eureka moments in my career came early that summer when Peter excitedly called me down to the electron microscope in the basement of our science building to show me the following image. This is of a cell that had its secretion blocked when the cells were warm to 37 degrees. Recall the image I showed a few minutes ago where there were only in a normal cell a very small number of these vesicles in the bud portion of the cell. Instead, in mutants such as SEC1 and in many other mutations that we obtained subsequently, the organelles intermediate in the secretory pathway fail to complete the process, but continue to be made. And now we have a cell, indeed a whole field of cells, where thousands of these vesicles accumulate, no longer restricted to the bud, but now spilling over to fill the entire cytoplasmic compartment. Well, this was for us a revelation. And so we excitedly, for the next year and a half, perfected techniques to isolate more of these mutations. We isolated then over 200 more such mutations. We're able genetically to define nearly a couple of dozen different genes. Mutations in each of these genes block the secretory pathway at a different step, each of them blocking the net cell surface growth of the cell. Well, as a result of then several more years of work, we were able to assemble a map of the secretory pathway that follows the rules that Palade had established himself for the pancreatic exocrine tissue, but which now applies even in lowly yeast, 2 billion years removed from human cells. A series of events from the ER to the Golgi apparatus into vesicles that fuse at the cell surface. But importantly, and quite distinct from what was possible in Palade's generation, we can describe for each step a series of genes and thus of protein products that are required to move, to create vesicles, to move them back and forth, to allow them to fuse, to understand at a molecular level the fundamental mechanism of this process and how it relates to the assembly of the yeast equivalent of the lysosome, or how the channel in the ER membrane is assembled so as to allow proteins being made by a ribosome to pass in the first instance through this membrane into the luminal interior of the ER. So we were very excited by all of these results, and the implications of these results immediately had influence on the biotechnology industry, which was beginning to grow in the San Francisco area. Because with the discovery that the yeast process was so similar to the human process, it became feasible to use yeast as a platform for the production of large quantities of clinically important proteins. So I helped a local biotech company engineer the gene for human insulin into a yeast cell, which could be harnessed then to allow yeast cells to manufacture large quantities of human insulin. And now, many years later, the pharmaceutical industry, including especially a company in Denmark, Novo Nordisk, manufactures human insulin by fermentation in yeast cells grown in huge fermenters shown in this factory. Fully 1/3 of the world's supply of human recombinant insulin is made by expression and secretion in yeast, relying on the evolutionary conservation of the genes. In addition, the vaccine against hepatitis B is made in yeast by introducing the gene for the hepatitis virus surface protein into yeast. The cells can be tricked into making little vesicles that resemble virus particles, but which lack the infectious nucleic acid. And when these yeast are broken open, these vesicles are isolated and used as a source of immunization. And some of you may have been vaccinated against hepatitis B infection, in which case you've received a dose of vesicles made in yeast cells. Now, as our work went on, and given the similarity between yeast and mammalian cells, it became feasible to consider how human diseases that affect the secretory process may involve the very genes that we discovered in yeast. And so we began to explore a particular part of the pathway that has a genetic defect known in humans that I'll describe in a few minutes. In the course of our further investigations of this process in yeast, we were able to show that there are two steps in the movement of proteins that are assembled in the ER membrane until they arrive at the Golgi apparatus. The first step is the cooperation of a set of proteins that are required to pinch vesicles from the ER and to collect secretory cargo molecules that are en route to the Golgi apparatus. And the second set consists of yeast proteins that are required for these vesicles to target and then fuse or merge by a process of membrane fusion at the Golgi apparatus. As we were engaged in these studies and having cloned these genes, my fellow Nobel laureate, James Rothman, also whose family came from Moldova, his grandparents came from Moldova, he managed to clone the equivalent genes from the human genome required for a membrane fusion process. And we discovered that we were, in fact, studying the same thing. The genes that we had discovered were equivalent to proteins that he had purified called NSF and alpha SNAP. We focused on these genes and proteins to discover the mechanism by which these vesicles pinch from the ER. We purified the proteins using a biochemical assay, and with isolated ER membranes incubated with these purified proteins, we could produce a population in the test tube of pure transport vesicles, all of a uniform size, coated with a fuzzy coat surface that we now know consists of molecules called COP2 or coat protein 2 complex, required in all eukaryotic cells for this step in the pathway. Now, work subsequently in other laboratories focused on the structure of the proteins that we had purified and discovered two sets of molecules, a complex of 3 proteins that bind on the cytoplasmic surface of the ER membrane and recognize and bind to membrane cargo proteins that are designed to be removed from the ER for traffic to the Golgi apparatus, and an outer layer, a kind of structure forming layer of proteins that forms a polyhedral cage that envelops a membrane with its cargo molecules and literally pinches it by a process called fission from the ER to create a transiently COP2 coated vesicle. Now, I tell you about this because after our discoveries, I was approached by a clinician, a pediatric clinician, a geneticist named Simeon Boygiev from Johns Hopkins University. And he had, in collaboration with a Saudi Arabian colleague in Riyadh, had found a Bedouin family with a rare mutation in this protein in the human genome. This protein called SEC23, a gene that we had discovered years earlier, but which is evolutionarily conserved, a mutation in an amino acid of this protein, which is invariant in all eukaryotic sequences. This particular mutation changes one amino acid to a different one and causes a seemingly subtle pathology that affects profoundly certain tissues in children that have mutation in both copies of this SEC23 gene. They have a facial malady, a misshapen head. They develop cataracts at an early age. They have brittle bones, but they survive seemingly normally with some deficit in spite of a mutation in an absolutely invariant amino acid residue of this protein. We obtained, in collaboration with my collaborator Leila Orci in Geneva, we obtained tissue samples from a parent who was a carrier having only one bad copy of the gene with no obvious pathology, and a tissue sample from one of the children who have the pathology. And by thin section microscopy, we evaluated the structure of the organelles in cells of normal skin cells from one of the parents, and skin cells from one of the children, with a remarkable difference evident in this image. The top panel shows a thin slice through a normal human skin cell with this membrane, the endoplasmic reticulum, ribosomes on the cytoplasmic surface, some granular material in the luminal space. The tissue from one of the parents who have a mutation in just one copy of this gene shows the ER to be somewhat misshapen to accumulate some granular material, but otherwise to not compromise the ability of these cells to secrete proteins. In contrast, a thin slice through cells from the skin cells from one of the children show a profound dilation of the ER membrane to a huge accumulation of granular material, at least part of which we've shown to be collagen. These cells harvested from the children accumulate an excess of collagen in the lumen of the ER because of a failure or a reduced ability to package collagen for secretion. Surprisingly, these children still mature, are alive, they're compromised, but they have a clear and profound deficit in the secretion of certain molecules. Well, in the course of this work, I had the pleasure, after Dr. Palade died, I had the pleasure of visiting Yale, who had established an honorific lecture in honor of Palade, who had established the cell biology department at Yale. And so one of my great proud treasures is this poster of the Palade lecture that I delivered at Yale, including the very handsome Palade in his prime. When my wife saw the poster and saw the image both of me and of Palade, she made a point of saying, "My goodness, Palade is very handsome, isn't he?" and clearly in contrast to me. So anyway, this is one of my treasures. And my host on that occasion was my great former graduate student, Peter Novick, who with his wife, Susan Farrow Novick, who had been in my laboratory, then became professors in the department that Palade started at UC San Diego. Well, as time went on, Palade moved to California, to the University of California, San Diego. And I had on several occasions the opportunity to visit with him including at a symposium in his honor near the end of his life, where we met and spoke. Palade was still mentally very sharp, but physically impaired. And it was great to visit with him on this occasion and to address the audience of his appreciative colleagues. I also met on this occasion and on many other occasions around that time with his wife, Marilyn Farquhar, also a very famous cell biologist in her own right. Regrettably, Dr. Farquhar passed away just a couple of years ago. And finally, let me leave you with the image of Palade from that symposium. Although age had taken its toll on him, he was as gracious and charming and brilliant as ever. And it was a delight from the moment I met him until the moment he passed away, that it was my privilege to have him as a close friend. I was asked after he died to write an obituary in his honor for Science magazine. And I remarked not only on his discoveries, on his brilliance, but also on his wit. And let me leave you with one example of the scholarship that he had that extended to a sense of humor in his role at a Cold Spring Harbor symposium on the subject of membrane traffic, where Dr. Palade was asked to deliver the concluding lecture after this symposium. And so let me quote from my essay. I'll also not forget his remarks at a 1995 Cold Spring Harbor symposium, where at the tender age of 83, he mused that according to the 17th century French literature, Cid Quiampeador, hero of the Spanish wars against the Moors, returned to court after a fierce and long battle, quote, long, but not as long as this symposium, as Palade put it, to tell his king that the battle ended because there were no fighters left. As the days of this symposium wore on, Palade hoped by the time he was scheduled to deliver his concluding remarks, the conference would be finished because there were no longer any combatants left to listen. So that was an example of his charm and wit. His knowledge of the literature and especially of Roman war history was really quite remarkable. Well, finally, life comes full circle. And now Peter Novick, who started that work in my laboratory so many years ago, moved with Susan Farrow Novick to UC San Diego, where they were then and are still. Peter is now the George Palade Chair of Cell Biology at UC San Diego. So with that, I'll conclude my appreciation of Dr. Palade, an amazing man, one who can never be reproduced, and his influence on the birth of modern cell biology cannot be underestimated. Thank you for your attention.
Dear Professor Randy Schekman, thank you very much for your wonderful lecture. And on behalf of the members of two academies, Romanian Academy and the Academy of Sciences of Moldova, to thank you for this lecture. On behalf of scientific communities of Romania and Republic of Moldova, to wish you good health and new achievements in your scientific activity. And we are waiting for you in the Republic of Moldova, in Romania. Please, as soon as possible, book the flights.
Next year, barring yet another pandemic or an extension of the war, I will be there.
Perfect, perfect. Thank you so much. And we are looking forward to welcoming you to the Republic of Moldova, to Romania, to the Black Sea region.
It will be my pleasure. Ian, thank you again.